the result of genotypes. If I were to summarize what genotypes mean the combination of alleles. And there are three ways you could describe combinations of alleles. For one, you can describe it as homozygous dominant. That's if you find two dominant alleles together. You can describe it as homozygous recessive. If you find two copies of the recessive allele. Last but not least, you can describe it as heterozygous. No need to include dominant or recessive. Heterozygous implies there are two different copies. Okay? These are the words and terms you can use to describe genotypes, not to describe phenotypes. Okay? Phenotypes are the characteristics. So how might we describe phenotypes? You may say that, oh, this P here is yellow color. My skin is yellowish brown. Yeah, these are phenotypes. Yes?
Then if there are co-dominant genotypes, like co-dominant alleles like the IA and IB,
Yeah.
then how would you describe them?
So if you have an IA and IB together, we don't have a term for that, you can still use heterozygous. Yeah. Okay.
How about how about
Or if it's two IB together. If it's two IA or two IB together, you could say it's homozygous dominant. Okay, because I understand, but it's still relative to the IO that we're looking at.
so there's no way to describe them differently.
Correct, yeah. I've not really heard of homozygous codominant. Okay, thanks for that question. Next, other than that, other vocabulary that we also learn, other than phenotypes and genotypes, we also learn that there are various ways that alleles can interact with each other. We see this interaction most when it comes to heterozygotes. Because if you were to have a big A and a small a, right, this kind of convention we use, we say that these two alleles exhibit complete dominance. Where the big A allele is completely dominant over the small one. Okay? So that's the first kind of interaction. Second kind of interaction that you have come across so far, codominance. This is if both alleles show their phenotype equally. So for example, if we look at blood types, we have IA, IB, these two are co-dominant to each other. Another example we gave last week had to do with cows. So for example, CW, CR, where R represents red, W represents white. Together, you get a lone cow. A lone cow is one with white and red patches. Okay, so that's co-dominance. There are many other kinds of interactions, but you will learn more in SH if you take biology. Okay, but for us, we need to know these two as the basics. Most of the alleles that we've been looking at so far, we've been looking at it in terms of the non-sex chromosomes. A lot of the puzzles we've been working truly with the 22 non-sex chromosomes. So I say again, for example, you have chromosome pair number 1, you have chromosome pair number 2, chromosome pair number 3, and so on and so forth, times 22 of this. Every time we solve genetic puzzles, we are truly only looking at one pair at a time. There's no way to draw a genetic diagram with all 22 pairs at once. Then you have too many crisscross lines we need to work with. So for example, if I just look at a particular gene with its alleles, very likely I'm just looking at one chromosome pair. This one could be the one with big A, this one could be the one with small a. If I were to work with blood types, I could be working with another chromosome. And blood type would be over here. IA, IB, and so on and so forth. Every puzzle you work with is really just one pair of chromosomes. Last week, we also delved into sex chromosomes. Because in reality, we do have genes found on sex chromosomes. And that is the 23rd pair. 23rd pair. On that 23rd pair, you can either have a X and a Y chromosome pairing, or you can have two X chromosomes as a pairing for the ladies here, right? And for males, XY. You do find that there are some genes that are linked to these chromosomes. For example, if I have a gene that's linked to the X chromosome, my convention to write it will be X, perhaps big N, and small n, where the caps and small caps represent dominant or recessive. The interesting thing about sex-linked chromosomes is that in the male combination, you only really need one copy of the allele for the trait to show. And this defies what we've learned so far, because we always learn that for recessive traits to show, we need it to be homozygous recessive. Yeah? Two copies of the recessive allele, then it will show. But not for sex-linked traits, because one copy is truly all we need. There is no contrasting allele to mask it, to dominate over it. And so that lies the danger of sex-linked genes. An example we learned last week had to do with hemophilia, the particular genetic disease that affects blood clotting. You find it affected more in males than in females, because males only just need one copy of the recessive allele. And a trademark sign that a trait is sex-linked is if you find that in the family tree, a large proportion of a particular gender has that disease. Then you know it's linked to that particular gender. Okay, and that's where we left off so far. These are the vocabulary you can use to describe all the puzzles that you need to solve. And we learned that on one hand, you can solve puzzles related to non-sex chromosomes. On the other, you can use it to solve puzzles related to sex chromosomes. Another word for non-sex chromosomes you may encounter is called autosomal chromosomes. Okay, that's a summary of what we've done so far. Today, we will do two things. The first one, we're going to complete more practice first. Once you're done with practice, you go through some of the questions. Because I find some of the questions here quite valuable. For example, this is the first time you're encountering a question where they ask you to explain yourself in words. Is this disease dominant or recessive? And you have to explain using the information provided. How would you do it? There is a very succinct way to explain. Alright, so we're going to practice some of these questions. When you see the word explain your answer using information, you often find in family trees or pedigrees that we provide numbers for you. That's because we want you to really make reference to these individuals when you're explaining. I claim that this particular trait might be recessive in nature because I noticed that when the individuals 7 and 8 have children, so on and so forth. So make use of the numbers to justify your answers. We also have another genetic diagram for you to practice. Okay, that's the first part of today's lesson. Then we'll answer some questions before we move on to the final part of the chapter, which is very, very, very short. And then we'll go on to our last chapter for your biology syllabus. Okay, so that's where we are today. Shall we do a little bit of practice? For those that are done with the practice here, you can move ahead to the next last part. But if you desperately need to revise your math, good, revise your math. Okay? to 8:35 to complete the questions, then we will be going through them. Those of you at the genetic diagram question, color blindness is a X-linked recessive gene. Yishuan did ask a good question for the genetic diagram to draw. Why in the world are you supposed to know color blindness is linked to X chromosome and it is recessive? Okay, so you don't know. I'm telling you, and from now onwards you're expected to know. Okay, so hemophilia and color blindness, these are two case studies that you're expected to know. If we give you a new scenario, we have to tell you, or we provide you enough clues for you to figure out. Yeah. This question, the color blindness, there's no way for you to decipher whether it's extinct or not. Kawa. Yeah, the Kawa is another example for codominance. Now you know. The Kawa and the as case studies. Yeah. Hey, give you a bit more time to work on your diagram. Finish, right? Sample size. Sample size. Last chapter is very short. Start. Oh, I see. I50, I'm going to go through all the questions. Today we will close this chapter. The question is exercise original recessive. Right, and then you say it's recessive, right? Oh, the answer is not here. Who is it? So that means this one is recessive, right? This one is no, this one has to be heterozygous, right? And this one has to be homozygous recessive, right? So I think that is you guys in your course. Yeah. You confuse yourself. Anyway, X is not a good formula. Oh, but the individual 6 is normal. Yeah. He said normally individual 6 is homozygous. 0. Okay, let's go through the questions. First question I'll go through is question 2a. Question 2a asks you, is the disease dominant or recessive? Yeah? And actually, we look across the board, a lot of you have chosen the right individuals to use as a justification. So which individuals do you use? If I scan across the board, many of us chose individuals 12 as well as individuals 7 and 8 as our point of explanation. So for example, Christine says recessive because individual 12 has the disease, comes from parents who do not have the disease. So the parents must be carriers of the disease, meaning that it is recessive. Okay, we scroll down, we see more similar explanations. Recessive, the disease was able to skip a generation as individuals 7 and 8 did not have the disease, but their offspring 12 had the disease. This implies that 7 was heterozygous and carrier, as individual 2 had the disease and was homozygous recessive. Okay? Look across the board, many of us chose these 3 individuals. So we are nearly there. I find that the way you are explaining it's not fully complete yet. Let me try to find one that I find is quite complete. The disease is recessive, as parents 7 and 8 has no phenotype of the disease, yet one child 12 shows the phenotype. This means the allele is most likely masked, therefore it's recessive. Okay, so this student over here, who is this? 1A, sorry, not 1A, Yi Hui says this LU is most likely masked, therefore being recessive, because she noticed that 7 and 8 do not have the phenotype, yet the child has it. So that means the parents must likely have one copy each, but it must be masked. So when you're trying to explain whether it's recessive or dominant, it is important for you to make relationship to what you observe in the phenotype versus what is on the genotype. For us, I think we are simply stating the individuals, their phenotype, and then we say a therefore. Let me show you a suggested answer. The individuals we chose are pretty spot on. So we look up recessive. Okay, second point. For individual 12 to be affected, he must have inherited one copy of the allele each from individual 7 and 8. Agree? Yeah? Must have inherited one from 7 and 8. That is how most of you conclude that they are heterozygous, right? But for many of us, we simply state that they are heterozygous or they are carriers. But how do you know that they are carriers? The evidence is in the child. The fact that the child is recessive and shows the phenotype, that means they have two copies, must have inherited one from each parent. Since 7 and 8 must carry one copy of the allele and phenotypically are normal, that means the allele is recessive. If both of them have this particular allele, yet they appear normal, that means this allele is not dominant one. And therefore, I summarize by providing a counter argument. Because if the allele were dominant, then you'll find that the parents must instead show the disease. So I think where we can add on one more sentence is to provide a counter argument. What if the particular allele that we claim to be recessive is dominant? Because if it is dominant, then the parents shouldn't be normal. The parents should have the disease. Okay? So I think we are pretty good at spotting the individuals to pick on, but now it's refining the answer. And therefore, actually, I have to say, if you ever encounter a question that asks you whether the trait is, the allele is recessive or dominant, most likely the answer is around the same. All you really have to do is to search for parents that do not have the phenotype, but produce a child that has the phenotype. That is the best justification you could always use, right? Regardless of family tree. And therefore, I do have to say, this answer is quite a template one. Okay? Because how you identify a recessive dominant individual is usually looking for such a combination of family, both parents normal, offspring not normal. Okay, next one. State the probability that individual 6 is homozygous for the hexa gene. So we look at these individuals here. So what's individual's 1 genotype? What will individual 1's genotype be? Heterozygous or homozygous? Individual 1. Hetero? Homo? Hetero? Make sense? Hetero? Okay, how will you explain it? If someone were to ask you to justify, why do you think individual 1 is heterozygous? How will you explain? Which offspring will you choose to help you justify your answer? Why 5, Vivian?
If the hexa gene.
So the question is, how do you know individual 1 is heterozygous?
The hexa gene is recessive.
Yeah.
Because of individual 12. So if this continuation from previous one, so the only way for individual 5 to have the disease is
Yeah.
if both parents had a copy of the hexa gene allele.
Okay, you see how Yuan Yuan is explaining? For individual 5 to have the disease, that means the individual must be homozygous for the recessive allele. To be homozygous for recessive allele must have inherited one from the mom, one from dad. Okay? Must have inherited one from mom, one from dad. So now we know for sure number 1 has one copy of the recessive allele. But because individual 1 is phenotypically normal, that means you must also have one copy of the dominant allele. Therefore, this individual is heterozygous. So I always make reference to the phenotype back to the genotype, phenotype back to the genotype. Okay, so individual 1 will have to be heterozygous. Individual 2, confirm recessive, homozygous recessive. Yeah? The offspring that we are looking at will be this way. These are the genotypes. So they're asking you individual 6, normal individual. What is the probability that this individual, individual 6, who is normal, is what? Homozygous for the hexa gene. Okay, what is the chance it's homozygous for the hexa gene? The probability will be 0. Because if you look over here, individuals that are normal, this person is normal, this one also normal. The only way for the individual to be normal is if they are heterozygous. So there is 0 probability that the individual is homozygous for the gene. Okay, so it's a 0 probability. Because the flip side would be that you have the disease already. Next one, C. What is the probability that the next child born to 7 and 8 is a girl who is affected by the disease? So there are 2 probabilities here to work with. We've encountered questions like that before, yeah? So how would we do this question? So let's scroll down. Okay, Heidi says 0.125. She put 0.5 times 0.5 times 0.5. Wow, okay. Heidi, how come you have 3 multiplications? How do you do this? 0.125 I think is the correct answer. But how do you do this? Oh, you don't remember doing this? Okay. 7, individual 7 and 8. If we go back to individual 7 and 8, I think Serene's one looks a little bit. Okay, she also arrived at 0.125. Both individual 7 and 8 are heterozygous. So individual 7 as well as individuals 8 heterozygous. And then they have a child. So for the child to have the disease, the child must be homozygous recessive. So there is a 1/4 chance or 0.25 chance that the next child will have the disease. So that's the first probability. Then she adds on another probability, whether it's a girl or boy. Okay, that will be half the chance, either girl or boy. You multiply both together, it will be 1/8, that is a 0.25 chance. 1/5 chance that it's going to be a girl with the disease. Okay, so that's how you work the probability out. How does Kelly do 0.5 3 times? I also don't know how you do. Sorry? You figure out already? Why you 0.5 times 0.5 times you do it?
Dependent is you
Oh, to get this one. Oh. I see, okay. Okay. Okay, so that is the next part of the answer. Then we go to your genetic diagrams, which is your bread and butter on this topic. We expect you to be able to draw a good genetic diagram. So I was walking around, you are really quite good at this by now. So let's look at Bates one. Why you laugh? Okay. So not enough information has been provided to you whether it is sex-linked or not. Okay, so I have to let you know, from today onwards, you're expected to know color-blindedness is not only X-linked, but it is recessive in nature. Okay? So, Bishan asked what are some case studies you know of so far? So you're expected to know color-blindedness. You're expected to know blood types. You're expected to know the lone cow, the cow one. Yeah? The cow, CWCR. So you're expected to know these case studies. This is not sex-linked. These two are not sex-linked, yeah? These are examples of codominance. This example of sex-linked. Another sex-linked one you're expected to know is hemophilia. Okay, so these two sex-linked case studies you're expected to know. Expected to know meaning that we will not tell you anymore which one is dominant or recessive. We expect you to know it already. Okay, these 4 case studies, we expect you to know which is dominant, which is recessive. No clues will be provided. Every other disease, we need to provide you clues. Sorry, I mean, not useless. Okay, I'm going to find another example. Let's look at. Oh, look at the right one. Okay. So if you look at the right one, okay. We look at the definition first. So you define at the very top. Let Xn be the recessive colorblind trait. Let X capital N be the dominant normal vision trait. We could say let X small n be the X-linked recessive allele. Let X with the big N be the X-linked dominant allele. Then she goes on to write all the headers. So I think a few headers are missing. All the content is here, but the headers are missing. Okay, the first header you should have is parental generation. Thereafter, you should have the parental phenotype and the parental genotype. Linear starts off with the parental genotype. From the parental genotype, you should have a cross. Then you should branch out to form your gametes. From here onwards, I think it's quite good. You find all the individuals with their genotypes. She then goes on to list down what each phenotype is. Then you'll find you should have a 2:1:1 ratio. You know you're correct if you get 2:1:1. Okay? You shouldn't deviate from that ratio. There is Madhu was very colorful. It's now like as I walk past. Okay? So you look at Madhu's one. Oh. Okay. So for Madhu, if you look at her genetic diagram, if I were to pinpoint the good things, actually largely she's followed all the headers. All the crisscross also is quite nicely done. You know where each one will go. By the way, you do not have to use different colors for any exam. Over here, put one more X over here, because if you want to do the, oh no, it's fine. Okay, what I see here, the phenotype is the father with normal, mother that's normal. Okay, understand? Okay, let's go to the phenotypes over here. So the phenotypes, I agree, normal, 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 colorblind. Okay, and indeed the ratio is 3:1 if you just look at normal or colorblind. So pay attention to the question. The question also asks for the gender. So gender plus the colorblindness or not. So when both are asked, actually you need to split up this ratio a little bit more. So this is a girl that is normal, a girl that is normal, a boy that is normal, a boy that is colorblind. So the ratio becomes a 2:1:1. Okay, so look at the question so that you know what kind of a ratio you have to look out for. Okay, so that's the genetic diagrams. Last but not least, you find this very last question over here. Explain why the observed phenotype typic ratio may not be as expected in this prediction. This is a very, very typical question you'll find. Even you go up to A levels, it's almost a template question. But this question is often asked because you want to see your understanding of genetic diagrams. At the end of the day, when you get this 3:1:1 or 2:1:1, these ratios, they do not really show unless you have thousands of babies. Okay? Sample size is too small for the probability to really show fully. Okay? So, if the number of offspring is large, then the observed ratio will be much closer. Or you can say the sample size of the observed offspring is too small, so it deviates from the expected ratio. This is a very, very, very expected answer also. Okay, it really has to do with sample sizes. And so the misconception here is that sometimes when you see the ratio, some students think that the chances of you, okay, look at these four, this probability over here. Sometimes when I ask students, this couple over here, they have 4 children. And then when I ask them what the identities, what the phenotypes of these 4 children are, they will say with certainty that the 4 children will definitely, 3 of them will be normal, 1 of them confirmed will have the disease. But that's not the case because this is too small a sample size. Only if you have thousands of babies, then you will see this probability show. Okay, it is a misconception to think that you definitely have 1 child with the disease. You could actually have all 4 children with the disease. You're just unlucky. But then if you were to have 1,000 more kids, then, maybe if you have 10 more kids, you find the other 10 will start to be normal. Okay, you just hit the 0.25 chance for all 4 kids. Okay, so we have come to the end of this particular section. I will take some questions for today before we move on to the very last section of the lesson. The last part of the lesson is very, very short. Okay, let's look at Zoe. Are there diseases linked to Y gamete? Y chromosome. Yeah? Gamete is an entire cell. Each cell would have 1 set of the 23 chromosomes. So, are there diseases linked to the Y chromosomes? Have. Then, are there dominant and recessive alleles for the Y gamete? Y chromosome? Have also. Okay? It's just that the example we've been working with so far, always we've been working with X-linked and recessive. But there are flipped ones. There are also X-linked, they are dominant. Y-linked, they are dominant. Y-linked, they are recessive. It's just that X-linked recessive, there's a little bit more of a puzzle to work with. The rest are a little bit more obvious. For example, if you have a Y-linked disease, confirm won't find it in females, correct? Confirm will just find it in males. Agree? Because males are the only ones that have the Y chromosome. So the puzzle becomes very easy to solve. If I ask, if a yellow and blue flower are cross-fertilized and the offspring is green color, is that an example of codominance? That's the question. Okay? If I go back to the cow example, where I have a cow that is red and a cow that is white, and let's say they are purebred. Okay, let's say if it is codominant, we end up getting roan, correct? Roan meaning there's red and white patches. If I apply this equivalent of thinking over here, then the cow will turn out pink. Agree? Yeah, the cow will turn out pink, correct? So the example you brought up, it's not that it cannot happen, but this is not an example of codominance. This is an example of a particular form of interaction you learn in SH called incomplete dominance. This is SH content. Incomplete dominance, that means when both are together, neither will show themselves fully. Both of them will show kind of like a half-half kind of thing, so they kind of blend with each other. So the example Ife is brought up here, this is more incomplete dominance, we get green. If the yellow and blue flowers were to exhibit codominance, you will get a flower that has yellow and blue petals together. Both traits will show. Okay? When the question asks to explain, are we allowed to draw genetic diagram to explain? Oh, okay. Usually, we just ask you to explain, and we just provide you 2 marks for it, don't draw a genetic diagram. Okay? So that is my reply to you. Genetic diagrams are usually worth 4 to 5 marks. It is like a biological drawing. Other than cross-fertilization, is there other ways to crossbreed? Cut off plant stem and stick it to another plant. Okay, what Ife has brought up over here, cut off plant stem and stick it to another plant, that's not fertilization, that's a process called grafting. I don't know if you've heard of this process before. It's possible, I've heard of a particular artist where he grafted many fruit trees into a single tree. So that throughout the year, different parts of the branch will bloom different flowers and sprout different fruits. It is possible. Plants are flexible to the extent that if you were to put a branch of one tree and stick it into another branch of another tree, if the xylems were to start to fuse with each other and they can still get water and nutrients, then actually the branch they just stuck in can still survive. But that's not reproduction. That is simply, that's what people call chimera. Yeah, when you join two beings together. Okay, so that's not fertilization. Serena asks, why are there different degrees of color blindness? Good question, and I am not sure. I'm not sure why. My first instinct is to think that environment may play a factor. Yeah, that's my first instinct. Second is that there could be more than one gene that determines color blindness. Except the one we teach you is just one of them. Yeah, that is my first instinct. Usually, if there are different degrees of a particular trait, like there's a range, it usually means that it's defined by a few more genes than usual. Because if it's only one gene, then you only have either one version or the other. But you are layering a few genes together, you have too many combinations. Okay, new cards. Tyson asks, how can a Y chromosome be dominant or recessive? Because every time you pass down Y, there's only one possibility. Oh, that's a good point. So can it be dominant or recessive? Hey, then my reply above didn't quite make sense. I guess, yeah, it doesn't make sense anymore. Okay, let me get back to you. Yeah, I may be wrong, or because there are possibilities when people get two Ys, but I don't think they will survive to maturity. Because dominant and recessive, I agree, it is a relative thing. Yeah, I don't know. Yes.
How do you get two Ys in the first place?
That's a good point, Mess up somewhere, one of the sperms is separate. No, it's also not possible. No, it's basically not possible. You're right. It's not possible to get two Ys. Okay, I don't know. Let's Google on the spot. Oh, by the way, if you ever see cats with like no tail, turns out it is related to dominance and recessive. You saw before? All this while in my whole life, I thought someone cut off the tail. Turns out it's a dominant trait. So if you see cats with no tail, they have one copy of an allele that results in a short tail. And I just learned yesterday that if you have two copies of the dominant allele, homozygous dominant, the cat will die as an embryo, will not make it out of birth. Okay, so let's answer the question. I have no idea which should research on the spot. Y-linked genes. Okay, is there dominant or recessive? Because need two copies. Okay, thank you. Then it doesn't apply. Okay, then I'm wrong previously. There are linked genes to it, but you're right. Recessiveness and dominance won't apply here. Because you only have one at any time. Thank you. Sion asks, why are pigeons male if they are ZZ and female if they are ZW and not humans? I don't know. This is an evolution thing. Yeah. Different organisms, the convention is different. It could imply that. Okay, for one, I have no idea what is the length of these chromosomes either, of Z and W. For all we know, Z is really short, and W is really long. Because I'm assuming that the female need more genes, right, to be able to carry out more functions. So I have no idea how much DNA is in a Z and in a W chromosome. That could be one reason why convention is different. Why don't humans that reproduce by splitting it? Okay, let me explain a little bit more what this means. Why don't humans that reproduce by splitting themselves apart in seeds? Please explain a little bit more. Like how come, like if I chop off my finger and put it in the soil, how come a few months later I don't pull out one whole inside that? Okay, why not? I think my best reply would be that all the cells in your body are fully differentiated or specialized. The only cell in your body that can still have the potential to become something else are stem cells. And stem cells, you find two areas you can find stem cells. One in your bone marrow. But even those stem cells are already not able to differentiate to all cell types. The only kind of stem cell that can differentiate to all cell types are the stem cells found in embryos. Those at the early stage pre-baby, that one can differentiate in every possible cell type. So if your finger cells are already differentiated to be finger cells, bone cells, blood cells, if I were to try to grow an entire organism from that, it's not possible because they've already reached a dead end in terms of what they can be. They cannot become any other cell. Even if they go through mitosis, you're going to get more fingers. Yes?
Let the growing child become.
Yes.
What?
Like me in the mom's.
No, like, I don't know, maybe an adolescent
Okay?
child.
Yeah, and then what will happen? You'll die lah. Okay, but why not, is it? If I take an individual, cut them down in the middle in half, why doesn't this individual go on to become two individuals? Okay, but it still goes back to the fact that every cell in your body is fully differentiated. They cannot divide and still become all. So you're saying if I cut down half, how come the brain of my one half doesn't mitosis to form another half of the brain, is it? Okay, when cells are fully differentiated, usually, for some of our cells in the body, they cannot go through mitosis anymore once they are fully differentiated. Yeah, so a lot of the cells in our body, we do have some cells, latent cells, that have not fully differentiated yet. They still can undergo mitosis. Once mitosis is done, then they fully differentiate into perhaps skin cells. So when I do get an injury, it is those cells that still have the potential to undergo mitosis that do. Then they differentiate to skin cells fully. It is not your skin cells that exist here that undergo mitosis that replace those lost ones. So that's why. And therefore, if you look at a lizard drop off tail, I hate it when lizards drop off their tail. My grandparents say if lizards drop off tail can go inside your ear and you become deaf. Anyone of your parents tell you that? No. So lizards drop off their tail, the reason why you can still grow it back because they still have stem cells there that can regrow an entire tail. Bone, everything from scratch. Some organisms can do that. So we are still trying to unlock the potential. Is there a way to direct our cells to become stem cells again so that they can regrow certain things? And that is actually a field of research.
How is it? Oh, is it because paper was very.
We look at tapeworms. Tapeworms, okay, they are simple, but more than that, tapeworms, they have a lot of stem cells across their body. Meaning they have actually a lot of undifferentiated cells. Cells that can still become any possible cell. So when you cut them into half, right, they activate those cells up. Those cells can then become become epithelial cells, blood tissue, so on and so forth. It's just that we do not have that. We don't have such cells with endless potential, unless in the embryo level. So how do plants have both male and female parts, but not most animals? How? If we elaborate on the question a bit more.
Like plants, some of them are like both male and female. But then in animals, it's like usually dichotomy.
Oh. So your question is why? Is it why do plants have both male and female parts but animals don't? I don't know, but if I were to speculate, the biggest difference I observed is that we move, animals move, but plants don't. So I wonder if that plays into it. If you don't move, maybe your chances of getting fertilized is lower. Maybe that would promote down the evolutionary line. It makes sense that you have both parts as a backup in case. In case you don't get a mate, I can still make it myself to get more plants. I don't know. That's my first train of thought because that is the biggest difference I see. It is in a lot of moving organisms that we find two different genders, right? But those that don't move, that we find single gender. They can find both genders in one. Perhaps. I'm not sure, but maybe. Okay. Okay, no more questions. Before we end off today, this segment is extremely extremely short. If you look at general population, for example, I just look at our class. Yeah? Actually, we are more diverse than we look. Truly, whatever we've been working so far, we notice we've been working with traits that have very defined characteristics. For example, we look at blood type, color blindness, hemophilia. It's a this or that. You either have it, you don't have it. You either blood type B, blood type O, blood type A, blood type AB. There is no broad range of traits. That is one category of phenotypes. Another category of phenotypes fall in the range of a very, very widespread. For example, if we look at things like height, things like skin color, what else? How big your nose is, right? Or how thick your lips is. All these if you look across our class, it's a spread. Almost like a bell curve, there's a range. Right? Just look at our class. If we look at height, these are the very, very tall ones, and these are the not so tall ones. If we were to line up our class in a row, I'm trying not to look at Nicole. I don't want I look at Nicole. I just want to plant here somewhere. Okay, but if we look at our class, right, we stand in one row, you'll find that majority of us lie in this region. Really, it's a bell curve, it's a range. And then here you find one like Keller, Jiaqi, right? Then the opposite side, you know. But it's a range of phenotypes. But if I were to segregate our class into blood types, you get a totally different story. So for example, who's blood type A here? Raise your hand. I have 1, 2, 3, 4. Okay, I have 4 blood type A's. Blood type B? 1, 2, 3, 4. 4 blood type B's. Blood type AB? 1, 2, 3. Blood type O? Okay. When we look at certain characteristics, there are distinct phenotypes. Some characteristics, there's no distinct phenotype, it is a range. And to sum up our lesson, actually there are two kinds of variation we observe in our population. There is the discontinuous kind of variation, meaning the kind of variation where it is not continuously varying. There are only a few phenotypes. And then we have the continuous kind of variation. Continuous variation. The kind of variation that has a very wide range of phenotypes. For example, skin color, height, so on and so forth. This is where we'll end off today. You watch a quick video before we close up.
Some kittens. There is a great deal of variation between species, but also there is variation within a species. One puppy is fluffier than the other. They have different coloured fur, and the kittens have different coloured eyes. Variation is the differences in the characteristics of individuals in a population. It can be due to genetics, the environment, or a combination of both. Let's start with genetics. Different members of a population have different DNA, unless they are identical twins. This means they have different versions of genes called alleles, and these alleles give rise to different characteristics. For example, on the eye colour gene, this kitten has an allele for blue eye colour, whereas this kitten has an allele for brown eye colour. If the species reproduce by sexual reproduction, the random mixing of alleles from both parents result in extensive genetic variation within a population of a species. This means that some individuals are better adapted to their environment than others, allowing natural selection to take place. For example, in a population of rats, some will either have better eyesight than others. These will be able to avoid being eaten by predators, so are more likely to survive to reproduce and pass on this advantage to their offspring. If you want to learn more about natural selection, watch this video. Some examples of variation in humans that are controlled by genes include eye colour and blood group. If you have a blood group A, it's because you have the alleles for this characteristic. This is genetic variation, and it's something that you were born with, and nothing can change it. Some variation is due to the environment or lifestyle of the organisms. Examples in humans are hair length. It depends on how short you decide to cut it, your accent, as this is linked to where you live, and if you have any piercings, tattoos, or scars. These are characteristics that you acquire throughout your life. Many characteristics are due to a combination of both genetics and the environment. For example, the height a person can grow to is determined by genetics. Tall parents tend to produce tall children. However, a person will not grow to their potential height unless they eat a healthy, balanced diet. Also, there is some debate on how much intelligence is due to genetics or the quality of education a person receives. Is intelligence more due to nature or nurture? To answer these questions, scientists can do experiments using identical twins. If the characteristic in both twins is the same, then it is most likely due to genetics. If it is different, then the environment has played an important role. For example, identical twins will have the same eye colour, as this is only due to genetics, but they will have different personalities, talents, likes and dislikes, and this shows that these are also influenced by the environment. So, in this video, we learn about variation and how it can be controlled by genetics, the environment, or sometimes both.
If you like the video,
Okay, I will pause here. Tomorrow I'll elaborate a little bit more on the two kinds of variation.